Does anyone have any questions they want to go over before we get going? Okay, I'll get going. And if anyone has any questions they want to go over at the end, we should have a good amount of time afterwards to talk about any questions remaining. So today we're just going to round out the visualization with a couple more visualization techniques and libraries. I think, especially with some of these, the first two methods, I think this kind of reinforces where R kind of shines when it comes to visualization. There are a lot of really good professional looking libraries out there for different functions that it really doesn't require too much manipulation to get a nice publication ready plot. So let's start with volcano plots. And I think this is something that everyone is probably pretty familiar with. This is a very visually convenient way to show magnitude of change and significance at the same time. And I think with some methods, like if you're just looking at full changes, you don't really know if that's significant. Say if you were making a heat map of full changes, that would be an issue. Or if you're using z-scores, that gives you some level of significance or how many standard deviations a measurement is moving, but it doesn't give you both in one go. And a reminder as well from some of our previous discussions that the p-values, it's really important that when you're making a volcano plot, that the p-value that you are displaying is appropriate for the platform you're using. So as a reminder, with RNA-seq, the data is not normally distributed, so you should not be using p-values from a t-test. And so you should be providing input from something like DE-seq. Whereas if you're making a volcano plot of proteomics and metabolomics data, in that case, using a t-test with some kind of false discovery rate correction, so adjusted p-value, that would be appropriate in those cases where the data is normally distributed. So with this, especially in this example that we'll be talking about, we're just going to be using that differential expression data out of DE-seq like we had before. And then we're going to be using this library available in the Bioconductor package manager. This is called Enhanced Volcano. This is actually a pretty recent package that came out that's really nice in preparing volcano plots with minimal input from the user. So again, here are how we prepared the data from DE-seq. So this is going through importing the data, performing the differential expression analysis, and then getting to that point where we got our results, or in this case, the ordered results by that adjusted p-value column. But I'm not going to output these results to a data table. I'm just going to keep them in memory within RStudio or whatever platform you're using. And then really it's a matter of just providing this data. So we provide the enhanced volcano function with that full data. We're going to tell it where to find the labels for the points. So here we're using those row names. So basically that row index for the different labels of these points. And then after that, it's really optional what the rest of these are, but we're going to tell it that the X is the log2 fold change column from the dseq output, and then that Y axis input is going to be not the p-value column, but the p-adjust, or the adjusted p-value column. And then I did some modifications to the fold change cutoff and the p-value cutoff, as well as providing a title. But then by default, you can see how this will label anything that passes the p-value threshold. In this case, it's blue. Anything that passes the fold change threshold, that's green. And anything that passes both is red and also labeled. So again, this has just been a carried over data set from before, but if you gave it gene name labels instead of these gene IDs, you'd have the gene names there. But in this case, I can't remember what the ID is for MCT1, but that should be one of these downregulated genes that are pretty significant. I think it's one of the top hits by adjusted p-value in the DESeq results. So if you want to read through the documentation for this tool, that is available here. And again, these authors did a really good job at walking through every possible example of different highlighting or labeling or formatting options. So reading through and seeing how they use the different arguments is very friendly to the user for making modifications. But one thing that I will note is that by default with these options, even if you use the p-adjust column, it's not going to label this as p-adjust. So that's something to pay attention to with all libraries, is make sure it's doing what you want it to. And if it's not doing something, figure out why, or if it's something cosmetic like this, go ahead and fix that for the final product. And so with this, you could probably output this as a PDF from RStudio, and then just go into the PDF in Adobe Acrobat and change this to p-adjust or something like that. So in that particular case, even if there's not a particular argument to modify that, that's something that you can do after the fact with different software. Are there any questions about making a volcano plot in R? Okay. So the next kind of common bioinformatics plot that I want to go over is the Kaplan-Meier curve. So these are, you've probably seen these before. So this will estimate the probability of survival as a function of a binary condition usually. So this is like if you wanted to know if a drug was having a significant effect on outcome, or does a genotype or phenotype improve the outcome. So basically, this is going to say at a given time, 1 through n, how many people are alive, and did they have condition A or condition B. So this again is available to make a publication ready plot. This is available through a series of packages. So this is through SurvMiner, which is available in base R, and then also Survival, which should be installed with SurvMiner. But then also the entire TCGA clinical database for all the cancer types is also available through this Bioconductor library. And that's something that I want to emphasize, especially with Bioconductor. There are a lot of public data sets like TCGA, even count data, where these are available as libraries, and so you don't need to curate the count table or whatever and import that into R. You can just install that library with the database, and then you'll be able to access that directly later on. Okay. So with this particular example, and I'll show you how this is formatted in case you want to use your own data. But here, just an example, and this is from this link here where they kind of walk through these examples. But with this is extracting the breast cancer and ovarian cancer clinical data. And then, so this would differ if you're importing your own data, where you need to kind of make sure that metadata is formatted correctly, but then you want to use this survival fit function. So fit the survival curve to the data based on, in this case, their vital status, so were they alive or not at a given time, and then the disease code, so were they a breast cancer sample or an ovarian cancer sample. And then the plotting, so this is built on another really useful package that I would look into, this called ggplot. So this one's called ggsurvplot. So basically a survival curve plotting package built on top of that ggplot plotting package. So here we feed in that curve fit, the data, and then just some things that I want to highlight here that were modified. And I think is really important that you include when you generate a survival curve is making sure you have the confidence intervals plotted, or in this case, you can just turn them on since this function will do that for you. I think that's important because there are several cases that I've run up against where without the confidence intervals, it looks like there's a decent difference between the two groups. But when you look at the confidence intervals, it turns out that they're actually overlapping. So in this case, you can see that survival is significantly different because none of these confidence intervals are overlapping. And then again, this particular library lets you have some control over labeling. So here I'm only labeling once every 1,000 days. You can change maybe the color themes, or whether or not you have this. This is telling you how many individuals are alive at a given time along this time course, I guess. And then, so say you wanted to use your own data for this. So with this, the information, it's really broken down into that. I guess there's a couple different ways you can do this, and it may need to be kind of massaged the correct way to get it to work. But you really do want just what is the group, so you can get that binary. Are they in the breast cancer or ovarian cancer cohorts, and then how many days basically. And I think that with this, especially in a lot of clinical data, one thing to be aware of, and I think this is actually combining this vital status with the time. So a lot of times in clinical data, maybe what you have recorded is only 1,000 days, but in this is just a binary variable where 0 should be alive, 1 is deceased. Maybe it's the other way around. I can't remember. It's probably the other way around. So 1 is alive, 0 is deceased. But you may have a case where this person, their time is only 967, but they're still alive. And so maybe a case where they were only followed up until 967 days. And so those people can be potentially excluded from the survival curve, or you'll have to see with this with this particular function whether or not that is being included, which I think with this vital status that will be factored in. Okay. And the last thing that I want to go over is this density curve. So I think these are a really nice, I guess you could look at these as somewhat of a, it's really just building off of a histogram, but this can be really handy if you want to compare two distributions from two different patient types, or maybe two different sequencing libraries. So this is kind of like a modified histogram, where instead of the number of, in this case, this is the distribution of survival times from that previous data set. So you can see most of the recordings are within the 500 to probably 1,000 range. But here, this is going to give you the proportion at a given metric. So here we create that density curve. So we use this density function. We can plot the actual line to outline that, and then we can fill that in with a given color. And I think that, I mean, you could really just have the two lines themselves from that density function. But I think it's really helpful to kind of fill in or shade the area under the curve with different colors if you want to compare two different densities. So in the second example, we can see that, so D1 is the ovarian cancer data, D2 is the breast cancer data. And then you can see that their distributions do differ to some regard. So you can infer from this that the ovarian samples survive on average longer, which, let's see. Which is actually strange when you look at this. I think that is where that follow-up data comes into play, where that's actually showing the opposite. So you may need to, in this case, clean up some of the data and make sure that's formatted correctly, or that you remove outliers or whatever. But this is done just like before, where you make one histogram, except for with this command, this allows you to basically overlay two different plots at the same time, so we can plot the second one. But then you'll notice that this also overlays the text. And so something that you want to do with these plots beforehand is make sure that they are scaled the same way, and that maybe you only provide the labels and title for one of these plots, and then maybe just the last one you provide the X and Y axes labels. So that's all that I have. Are there any questions left over from basically anything? I'm not planning on holding any review sessions on Friday. So if you have any questions about command line stuff or anything else, we can go over that now with the remainder of the time. And if you have any other questions afterwards, feel free to reach out to me whenever. I'm happy to help you troubleshoot anything this week or next year or whenever. So
Hey, Jordan.
yeah.
So I have a question, or actually a problem that I ran into regarding DE Sec 2.
Okay.
So it's like in our studio when I start with it, and like initially we had to read tables, like first the count table and then the sample table.
Uh-huh.
So my count table read goes fine. I can do the head function, see it, but when I try to do the sample table, I am not able to do that. There is some error coming, so I don't know, is it related to the table I have or something? If you want, I can I can just put the Slack snapshot for that.
Yeah, that'd be great.
Yeah,
I can
just
go back to that slide so we can compare. Maybe there's just like a formatting
yeah,
difference.
just a second. So do you want me to do it in like the Zoom window or the Slack channel?
Can you attach it to the Zoom chat?
Sure, just
Okay.
a second. I will. Zoom chat. Let me find it. Desktop. So this is the sample table, the screenshot that I got in our studio.
Okay.
And if you want, I can also upload the table that I am attaching. Maybe I think that because I took the metadata table directly from the website, like the SRA, from the SRA run table, like the metadata attached to it. Maybe I'm doubting it's something to do with the data table too. Let me just, I'll attach
Okay.
the data.
Okay. So at your separator, the -T. So if you look at your second quote, the formatting's kind of weird. I think if you just retype those quotation marks.
So just a second.
I can just
I'll
share my
just
screen
open
to
my
you.
PNG.
So if you see on the screen, so and it's just like a really minor thing where for some reason the formatting got messed up in the copy between for this particular character.
Okay.
So if you change that, I think that should fix the problem.
Okay, right before the backslash. So the second one after the backslash, right? So,
Yeah,
okay.
so right before the parentheses.
Yeah, I'll try doing that and see if it works for me.
Okay.
Because when I do I get these plus plus plus. What does that mean? Like?
So that's just saying that it hasn't recognized an end of the command that you're typing. And so it's just saying this is a new line, but it's still part of the previous function that you're typing. So it's all going to execute as one.
Okay, so I'll try to fix this thing and see if I can get it work. So
okay.
Yeah, so yeah, this is the I have just reached till this step, so I don't know. I have this question. So you said like you won't take any questions, like as we won't have any review session on Friday. So if by the end of this week or during the weekend, if I come up with some question, can I approach through Slack or?
Yeah, definitely. So I'll keep the Slack channel open for a while.
Okay.
And like I said too, if you ever have any questions, next week, next month, next year, whatever, feel free to reach out to me. I'm more than happy to help. And even if you end up running your own sequencing analysis or something and you just want me to read through your code really quick to make sure it looks okay, I'm happy to do that as well.
Okay. Yeah, thank you. That's nice. And one more thing, like, so I kind of probably troubleshooted for this, like, but when I was trying to install Bioc Manager, and I was going through the initial commands that, like, through install.packages or like, the other things that you showed in the slides before, I wasn't able to do that. It was always giving me that Bioc Manager is not compatible with our version 4.0.0. But when I went through, like, tools and just in install packages in window, I just put Bioc Manager and I did it. I was able to do that.
Okay.
So do you know why I wasn't able to do the other way?
That is a really good question that I don't have the answer to. I guess the best answer I can come up with is programming magic. Sometimes, for whatever reason, things just work one way or they don't work the other. Yeah, sorry, I don't have a good answer for that.
That's fine. It's just like I thought like maybe I was doing wrong, but it was just saying it's not compatible with our 4.0.0. I don't know why.
Yeah. I mean, maybe that with that install packages tool dropdown thing, it could be able to do more stuff behind the scenes to kind of work through incompatibilities. But yeah, again, I'm not really sure. So.
Yeah, that's fine. Great. Thank you, Jordan. Thanks a lot. Yeah.
Hey, Jordan, should we post that, or should I post something about the making Conda strict channel?
Yeah. Do you want
to? Yeah, I can post something. I don't know. I guess.
Do you want me to just copy and paste? Oh, sorry.
But then I should, put other people. Things they might want to know too. I usually just assume it's something I've done. So.
Okay. Did you want to go over that right now?
Sure.
Okay.
I tried at work, but I couldn't even remember how to log in on an Apple, so I did something else.
Okay. So, do you want me to just kind of walk through it and? In a video conference.
Yeah.
Okay.
I'll put you on. I'm gonna mute because my wife's having an appointment. So.
Okay. Sounds good.
So you can just go.
Yeah. Sounds good. So, let me log in real quick. Okay. So the question was, and you can correct me if I'm wrong, but so there's an issue where when trying to run SAM tools, it basically ran into this like dependency clashing issue where there's a particular library that SAM tools depended on, where there's an incompatible version. And I think this is the OpenSLL library. So I think this is a library that it's kind of like not a bioinformatics package. It's used for, I think, like data transfers between servers or something like that. And so, well, I think that package is available on Bioconda, but that version on Bioconda is incompatible, and it's been incompatible for quite some time. And so this is an issue where you need to make sure that that package is downloaded from the Conda Forge channel instead. So I'm going to go through the steps for doing that. So say we were to create a environment. I'm just going to call this new environment. It's going a little bit slow. Uh-oh.
I got the impression it was important, the order that they're done. Like,
yeah, that's
set the channels before.
Yeah, so you basically, it kind of stacks the most recent one that you called on top of the other.
Mm-hmm.
Yeah, so I'll just kind of walk through those commands. For some reason, Conda's not happy on my supercomputer right now. So you'd make a new environment and activate that. So it's a activate new environment. I'm just going to let that fail. So then you want to, before you start downloading anything, set the channels. So this allows you to not specify every time you try to install something. It just knows, here's the first channel I check, and then here's the next one. So if we want to make Bioconda accessible, we would do that first, because when it comes to that OpenSSL or SSL library, we want to make sure that it grabs it from Conda Forge and not Bioconda. So by specifying the second, this will move Conda Forge to the top of the priority list. I guess that's supposed to be channels, plural. So now whenever I install all the packages for Conda, it'll first check, is this available from Conda Forge? If so, download it from there. Okay, maybe it's not there. Now I'll check Bioconda and so forth. So I just make sure that Conda Forge is the priority there. I think, I forget if you can actually, I don't know if you can list that. Yeah, that's how you would do that. And then that should solve any of those, at least the issues with star dependency and compatibilities. That will solve that issue.
So this is different than making the channel strict. This is just.
Yeah, so I think that for whatever reason, and I don't know, I wasn't able to figure this out, why it basically won't let you make it strict. It's only flexible or default. But with this case, I think what the flexible is doing is, say you have two channels, but maybe the lower priority one has a newer version. I think it would install the newer version.
Right.
But I think they've just stopped updating the open SSL library on Bioconda. So it's always going to have a more recent version on Conda Forge.
Mm.
So I think that at least in this case, the flexibility of the channel order shouldn't be an issue. Did you run into any issues with that after you set the channels, or?
I haven't done it yet.
Okay.
I'll try that here in a minute. But yeah, I can post questions I have too on Slack. I think other people can benefit from my screw ups.
Sounds good. Yep. Don't be afraid to ruin the code more and more, because that's the only way to figure it out.
Yeah. I usually will try to figure it out on my own a few times before I'm like, I give up and go to Sensei. Yeah. I'm like, I don't know what's happening. But I feel like if I really try on my own and can't get it, then,
Yeah.
you need intervention.
Yep. I am more than happy to intervene.
I appreciate it.
I am willing to save other people headbanging against the wall for
Yeah,
helping out.
but I think that we all have to go through a certain amount of it.
Yeah.
I'm hoping if I just force myself through it, I can get a lot of the head banging out of the way.
Yeah. Yeah, but I think that's one lesson to take away is even I don't have all the answers and I don't understand how everything works. And sometimes it's just a little bit of blind faith that,
Yeah.
okay, it didn't give me an error, so I'm going to go with it.
Well, all right. This was very helpful. Thank you so much.
Yeah, no problem.
Hey,
I'm sure-
Jordan.
Yeah. Oh,
I
Sorry.
was just going to say, I'm sure I'll talk to you soon again. That was all.
yeah. Sounds good.
So it ran, like I fixed that and it was fine. I could get the sample underscore table like header run.
Okay.
So yeah, thanks.
Yep.
So, but I have another question in this same command. So in this you have warn is equal to false. Why is that?
Let me go back to the slide real quick. Oh. Nope. Warn equals to false. Good question. I don't remember. I think basically what this does is if there is an issue, it just reads through to the next line and skips that line.
Mm-hmm.
You can probably set it equal to true and it'll be fine. I think it's just when I originally used this code a year ago,
Mm-hmm.
the example that I saw that I based my code off used that option. So, I mean, it's a thing where a warning
Mm-hmm.
in a function will always continue, but it'll just print out the warning, whereas an error will stop the code from running.
Mm-hmm.
So I think whether or not you have this as true or false, you're still going to get an output.
So
But
it's to tell it like even if there is a warning or something, just go past that.
well, so this is kind of a verbose versus non-verbose thing. If you don't want it spitting out a bunch of warning stuff, and it may be a case where I saw warnings that I didn't actually care about and didn't actually matter for what I was doing.
Mm-hmm.
And so I just suppressed the warnings so I didn't have to deal with seeing them printed out every time.
Okay, got it. Yeah. Yeah. And so, yeah, I printed out the head of my sample table and it looks very weird to me. Like, I think it's a formatting issue or something with
Okay.
that. I'll try to fix it myself and see, but if I cannot, then maybe I'll just mail you that table file
Okay.
so that you can let me know what's wrong with that, because that's the data set I directly downloaded and used it.
Okay.
So maybe it has some issues. Yeah.
Yeah.
That's it.
I would take a look at the table in the slides folder on GitHub
Uh-huh.
and just look at the formatting there, and maybe you can identify some
Yeah.
differences.
Yeah, I'll try to do that first myself.
Okay.
Yeah, looking at the table that you have uploaded
Okay.
on
Sounds
GitHub.
good.
Yeah.
Yeah, let me know if you're still having formatting issues and I can help you.
Sure. Thank you.
Yep.